Thank you very much. And it's really a pleasure to be here and present the new data and give you also a glimpse that you can use Aptenas not only for detection but also for control of gene expression. And after long years, I really changed my introductory slide and used this nice picture from Yannis, who will give the talk afterwards. Because if I don't present for an audience that is aware of RNA, what RNA can do, and so on, they do not really know that RNA indeed can really form such beauty and beautiful structures that we all like, and that RNA or nucleic acid can then form binding pockets and recognize proteins or can recognize more molecules. So that's why I changed to this nice introductory slides. And then I always explain, and I don't have to do this now, what SELEX is, and that with the selection you really can end up with this beautiful, nice guys here, the aftermath, and so on and so forth. And I can skip this, of course, now. So, and then, of course, I give the introduction what aftermath we all can do, and we will hear about many of these topics. But in the next session, we will focus on this part here, how you can apply aftermath to control gene expression and to use them as molecular switches. So we have 3 different ways how you can use this aftermath. One is a kind of roadblock mechanism. Here you simply take your aftermath, put it in the untranslated region, and then it's a roadblock where the ribosome comes and bumps into the aftermath, but only if the ligand is bound, because helicase activity in the ribosome allows the ribosome to open up cellular structures. And this is also the reason why this approach works pretty nice in yeast, but it fails up to now in the human or in the higher eukaryotic systems. So this is one, control of translation initiation. The other is control of splicing. You know, splicing is the major source of the diversity of our proteome. And you can use the aftermath to control if an intron is spliced in and out. And this works also pretty nicely. And it works, this time it works in yeast, but it also works in the human system. And I will give you 1 or 2 examples later on. And the third strategy that really works out nicely is that you make use of the small RNAs. They are called ribozymes. If they fold correctly, then they can cleave itself here exactly at one position, or also shown here in the secondary structure. And you can control the activity of these ribozymes by attaching an aptamer onto it. In this case, the strategy is you need here this loop-loop interaction, and then you can disturb this loop-loop interaction if you have an aptamer attached to it. So these are the 3 different ways how you can apply aftermath to control gene expression. We will hear, there are some other strategies. Eric, you give a talk or you have a poster? You give a talk afterwards. So we will, after the lunch break, we will have another excellent talk about another strategy, but in our lab, we focus on these 3 ideas. So in the meantime, I would say there's are not longer proof of concept studies. So in the meantime, these really works so that you can apply them. And here's only one example. It's a lipoxygenase, and here we have a kind of suicide exon. It's a completely synthetic exon, and we control with the aptamer if it's recognized or not. And the system is designed in a way that if the aptamer or if the suicide exon is included, then you have 7 stop codons, and this leads to to a stop of translation or to non-mediated decay. So you don't have any product, but if you add tetracycline and it's skipped, then you have the green exon here fused to this green exon here, and you have the correct message ending up in a really pronounced gene expression. And this works really nice, and you simply have to take this part out and you can put it into any of your target genes. So it works with OCT4, with Merck and Marx and CD20 and so on. So it really, I would consider it in the meantime as a robust tool to switch on and off gene expression. One example was here, a colleague of mine, he was analyzing B cell development and the role of CD20, an important receptor on B cells. And he integrated our suicide device into B cells by CRISPR-Cas into the endogenous loci, and then he could plus minus tetracycline switch on and off the expression of CD20 on the surface, and it really worked in a really completely way. These are the examples, and we are really happy in the labs that we see that our tools can really applied to help to decipher the function of CD20 during B cell maturation. So, summary slide for the first part. We consider ourselves as someone who develops tools. We are independent of the organisms, so we work in all of them, and we have different mechanisms. And for what? To control gene expression. So these tools are really cool because they have a very small metabolic burden. There are RNA only. There's no interference of proteins with immunoreaction. Small and portable. We currently work that we also can manage isoform switching, so we have really good progress here. And an aim is also that you can apply these, for instance, for metabolic sensing if you want to optimize strain development. So you have to sense if a certain metabolite is there or not, and can do screening for better strains. So, but for this you need, of course, not only two aptamers. So currently, if you look into the literature, everyone is working with theophylline, some with, we working with tetracycline aptamer. So there are only a few of them, and of course, I talk about this for years now, that my lab is interested in finding new aptamers that you can use for riboswitching, and because majority of small molecule binding aptamers can't be applied. They simply do not work. And a pure PhD student in my lab tried many of them, failed many of them. And so we tried for years now to find new riboswitches. These were the first guys we were working with, neomycin, ciprofloxacin. If we do a normal Celex, we end up, of course, with aptamer. They all bind nicely to the ligand, even in the low nanomolar range, but the majority of them fail. So we have an in vivo screening, and with this in vivo screening, we can find, oh, here I have the same problem. We can find new aptamers that regulate gene expression. One guy is shown here, neomycin. Another guy is here that binds ciprofloxacin. And then also these are all data, but together with Jens Wöhnert, he looked into the structure and he could show that for our neomycin aptamer, we really need conformational switching, conformational change to allow gene expression. And we think that this is a very important and critical point, because most of the aptamers, they are already preformed and the ligand comes and docks on it. You have excellent binding and they do nothing in vivo in the cell. So of course, if you want to block something, you don't need switching. If you want to use it for diagnostic, or so you also don't need the switching, but you need the switching if you want to apply the aptamer as a switch. So, and this was the point when we thought, okay, then maybe the normal Selex is not sufficient. We need, yeah, a kind of a changed Selex, and most of you will be aware there is the capture Selex, and capture Selex is a selection where we do not only select for binding, but also for switching. So you have, in this case, coupled your pool to the solid support, then you add your ligand, and you elute only the ones that bind your ligand, but also they have to somehow dissociate from the solid support. So you somehow select for binding and conformational changes. So we did this, we found a new guy. This is a paromomycin aptamer, and here we could nicely show also the high specificity. If we compare paromomycin with neomycin, they differ only by one moiety. So neomycin has an amino group here. They have a hydroxyl group for paromomycin. You see, very specific for paromomycin, but not the neomycin, and vice versa. So this was the first proof that this was we find aptamers with capture Selex, and then we did this calculation here. So we did deep sequencing, and we were looking where we could find our aptamers, and we did a sequencing of the cyclofloxacin Selex, and here we could find our riboswitch not among the top 1,000. And this was completely astonishing for us. And we have a really nice and rich Selex pool. We look for our aptamer and we find it, not among the top 1,000. So we really were lucky finding it, and it was much better for the paromomycin. And now I have to speak up. We repeated it. We could find a tobramycin riboswitch. Here, again, we found this, we shortened it, and then we could show that it has a nice binding behavior, but the two-step ones, and here, again, together with Jens Wöhnert, he could show that we have two binding sites, an upper and a lower one, and we could dissect it. We could really show one aptamer could bind the ligand, the other aptamer could bind the ligand. And then we introduced point mutation, and we could kill activity. If we introduced here in the lower part, we did not kill activity, so we retained aptamer. If we inactivated the upper part, this shows that unfortunately we did not have a covalent binding, but it also shows that we even we can shorten it much more. We can simply delete it. We retain activity, and we ended up here with this tobramycin aptamer that nicely react to our ligand. And then Jens were able to solve the structure. And here you see, it's an A-form helix, and the ligand simply flips in, binds. This was the inactive one. And here we have the active one. And here you see this nice architecture where you have this kink in the backbone, and you have a very specific contact here of this base. And you can see this here to this additional amino group, and this also shows the high affinity and also a kind of discrimination between kanamycin A and tobramycin, because here the amino group is only here in this blue and the green compound, not in the red. And this is this one contact that's missing. And then finally, in the last minute, I was always asked, is the capture Selex really better than the conventional one? And then at a certain time, we decided we have to prove it. new ligand, doxycycline, again, a Selex, and our in vivo screening. Yeah, this is important to find, and the in vivo screening really finds the regulating abdomen. And here, Selex, okay, in vivo screening, yeah, here. This was the in vivo screening of several rounds of the normal conventional Selex, and this was the in vivo screening of the capture Selex. And you see that we really could find regulating abdomen. But, and there's always a but in biology, here, one outlier. And finally, we have now two doxycycline binding abdomens. This is a good one, comes out of the capture Selex. I will not talk, will not talk about this. We have here the outlier, and this is an exceptional good abdomen. We found it by chance, and the next talk given by Janis is about this cool little guy. Yeah, and here simply, if you look into the Selex, here you have a lot of clades who evolve during the conventional Selex, and here with the capture Selex, our cabby, we call it cabby, took over and is the dominant species. And this was really a proof that with the capture Selex you enrich the regulating abdomen, but with the conventional you also can find them. And so we have now a slew of different abdomens in our lab. And I have to skip, unfortunately, the second part where we somehow put them together to increase on the one side, the dynamic range by simply taking one abdomen, putting it on top of each other. If you are interested in doing such things, there are some design rules, but in principle, you simply can play Lego with them. You can find some rules here in this publication, and then we try to find, with the help of artificial intelligence, one where we really could put them together to design an AND gate where we have the response to only if both ligands are present, then we can switch off gene expression. And this was also possible by simply randomizing this to connected parts. And with this, I'm at the end. Thank you for your attention.